Thank you, Keller. So I hope that everyone still have some space for single-cell proteomics because my talk would be full of them. So I think that at this stage, you all understand that at PTI, we are really interested in understanding aging because aging influences the biology from neurodegenerative diseases, as Meon just introduced to you. But in the immuno team, we're really interested in how aging impacts your immune system. And there is a lot of things impacted by aging, such as the organ evolution, so you would produce less immune cells, loss of proteostasis, a strong immune unbalance over time, immune cell senescence, a lot of your immune cells will die, as well as inflammation. And there's many consequences of all those aspects, but one of them is a global immunodeficiency. And I think that a concrete example of that we have all been through in the recent years is how our immune system answers to infections, either against SARS-CoV-2, which is the virus responsible of COVID-19, or influenza that you probably all have had during the winter because it's a seasonal flu. And if you look at this graph of mortality rate as a function of your age, you can see that the older you get, the higher your chance of dying from infection is increasing. And it's a simple fact that you're more vulnerable to infections when you get older. Fortunately, when you get older, usually you also get wiser. So when you look at the vaccine, the percentage of vaccinated people, when you're 65 years or plus, when it comes to flu vaccination, about 70% of the people are vaccinated as compared to 35% of the younger individual. However, vaccine fails to protect them efficiently, as when you're really young, the vaccination against flu protects you in 60% of the case. When you get older, it can drop as low as 10%. So not only you are more vulnerable to infection, but also when you get older, that vaccine protects you less efficiently. And so in our team, one of our projects is to try to understand how immune responses are impacted by aging and how vaccine responses are impacted by aging. And to do so, we started a collaboration with the CBC on flu vaccination to get access to a really incredible cohort of patients. And so far, we only screened 4 of them, 2 old and 2 young, that had been vaccinated against the flu. And we collected their blood at before vaccination and up to 28 days after vaccinations and isolated all the immune cells or PBMCs. And we decided to apply single-cell proteomics not only because we're a proteomic institute, but because there were a lot of reasoning behind that. First, the need to zoom in at a single-cell level because not all immune processes are impacted evenly by aging, but also within each immune cell population, you have a strong heterogeneity on how they're impacted by aging. So we need to go down to a single-cell level to be able to get some answers. And also, as you're probably familiar with that, RNA abundances do not translate well with protein abundances, but also by looking at protein, we're hoping to not being only able to look at protein expression, but also look at protein modification and also protein-to-protein interactions. And to build all this analysis, we decided to do a single-cell proteomic pipeline that rely on nanoproteomic sample preparation or NPAP developed by Andrew Leduc at Northeastern. Then to run all of those cells using purified single-cell proteomics or PSTOTs that has been developed by Ralph at Northeastern University and do induction biological analysis. And to give you some sense of numbers of what we have been able to do on this project, we are currently able at PTI to prepare about 2,000 cells per day for a total of 20,000 PBMCs that has been prepared in only 3 weeks. And all those cells has been acquired on an Express 480 in less than months. And also because we love challenges, most of you know LSL, for instance, doctors as Meon talked about. But when it comes to immune cells, they're really, really tiny. So the amount of matures that we're working with is like significantly lower than most of the cells in the human body. So to go through all the steps, we first decided to do NPOP by combining TNT, which is an isobaric tag. One of the main reasons we use them was for the capacity to place up to 32 samples together, and we hope that PSN tags will allow us to do the same thing in the future. And we use the 7-1 developed by Sayenians to do single-cell preparation. And we have optimized it thanks to the hardware of linear in order to be able to develop those solid duplex array, as you can see in the picture of the slide right now. So we were able to isolate cells individually in an repet to lysate, digest them, and also to tag them before doing a single cell proteomics. Out of those 20,000 PBMCs that we collected, some of them were cell debris, some of them were clump of cells that just wanted to join the party, but most of them were really nice immune cells that we can analyze and notify. And so after some filtration to only keep the proper cell for our analysis, we got about 17.5 thousand PBMCs to be run in our mass spectrometer. And then with the help of Sarah, we decided to perform PSCOP. And one of the questions that maybe you guys had is like, why PSCOP and how did we do it? Because there is many ways to analyze cell by proteomics and single cells by proteomics. And one of the main reasons is because PSCOP allows to analyze thousands of protease peptides across all the single cells. And also it maximizes instrument time. And for us, it was super important because of the huge number of cells we're dealing with to be sure that we would have a really high sensitivity and a really high data cut weakness, as well as the best protein coverage. And PSCOP allows us to have like an increased protein coverage as compared to other techniques. And in a nutshell, PSCOP, as the name stands for, rely on building a purity list. And to do so, we chose the data-independent acquisition of the samples to build a purity list and to collect the retention time of each peptide to allow a software called Max Quantlife to know when to prioritize what peptides over another one. Then we did a couple of scoots using data-dependent acquisitions to be able to refine this list and to only keep pushing this with the peptides that we call efficiently prioritize. And then we finished by polishing it to only conserve the peptides that we were seeing consistently within all our single cells. And we took some decision making in building these priority lists because, as you can sense, a priority list means different tiers. And so how did we define them? And the first one is because I'm biased in the immunology, the highest tiers contain all the proteins that belong to immune-related functions. And then we disperse the remaining peptides into 3 tiers based on their overall intensity. And also to maximize our chance to detect all those peptides, we increase the maximum injection time in our mass spectrometer for these higher tiers containing all the immune genes to consistently detect them. And also we get a little boost for the low avalon peptide to also be able to acquire them as often as possible. In total, we got about 1,100 proteins, with 20% of genes being on the higher tiers, that was the immune genes tier, 20% of them in the medium tier, 40% of them in the high tier, and 10% of them in the low tier. And how this looks like in practical, Max Quantlife, every single time that we see a peptide, we decided it would be sent for analysis in our mass spectrometer. And in red, you have the detected one. In green, you have the sent one. And you can see that for the lower tiers, only in 50% of the time, it will prioritize them. When it comes to the medium tier, it's 90% of the time. And when it comes to the higher tier, every single time you see a peptide, it will prioritize it and send it for MS2 scaling. We did some quality control on our sample, and here you have the graph of the sum intensity of the peptide per single cells in blue, and in how negative control in red. Negative controls being just droplets that did not receive any cells to look for the background of our analysis. And out of that, we filtered out about 30% of the cells that didn't match our quality standard, and we ended up having about 14,000 PBMCs that we analyzed for biological functions. It's led sweating about 800 PBMCs per conditions and 3,000 PBMCs per patient. And as you can already see, for example, in the green side, we had to deal with a lot of variability in sample qualities, which is the things that always happen with primary cells acquired from patients. In total, in those 48,000 cells that we collected, we have consistently about 2,800 peptides per immune cells, and about 950 proteins per immune cells, from which 240 of them were related to immune response size. And after imputation, we got up to 1,600 proteins per human cells. And then, and this is really preliminary, we started to try to solve a puzzle with Megan that was what cell type was present into our sample. So as compared to what Yaron presented, where he was working in the pure population of neurons, PBMCs are actually like made of like 5 to 10 main cell types and a total of 25 to 35 different cell type. And we had no way to know what cell was this in what droplets that receive what tags. So to solve this huge and troubling puzzle, we decided to leverage a single-cell RNA-seq dataset published by Aoned and All in Natural Communications that basically did the RNA sequencing of 1.3 million PBMCs for 120 patients. And we used this huge dataset to try to align our single-cell probability data and try to cluster our cells and being able to level. To do so, we first selected the most highly variable gene on this RNA-seq dataset out of the top 3,000, and every single of one label here warning to immune responses. Within the cell, the same from our single-cell protein dataset, and we got also like a couple of immune genes. But when we looked at the gene in common between both, we had about 240 genes that were highly variable and in common. And we used those genes to do clustering first of the RNA-seq data, and we're able to cluster cells in like 8 clusters, and to actually correctly label them with the main cell type presented to the PBMCs. And we did an asymmetric clustering of our single-cell proteomic data to try also to map them and to attribute them a cell type. Interestingly, we saw that overall the distribution of the cells in the different clusters was pretty similar, as the exception of the clusters 3 and 5. It can come from the fact that we work with samples at different vaccination stages, so it may have changed the cell population distribution. Also, 3 and 5 belong to the same lineage, the myelocyte, and maybe we are just not able to correctly cluster them with our front approach. So it's something that we're working on right now. But interestingly, we went one step further and we tried to confirm that the gene overexpressed in those clusters has some biological meaning. And we were first really happy to see that all our cytotoxic lymphocytes were clustering together, but also that they were overexpressing granzyme and porphyrines that are the two most important molecules to trigger the functions to killing cells. Also, it was really interesting to see that CCL5 was the main driver of separation between NK cells and T cells, and CCL5 is a CD3 markers of effector cells. We also look at the monocyte cluster, and we're able to see that CD14, which is a monocyte marker, was overexpressed in this cell population, as well as lyse, which is an important enzyme to play a role in fighting bacteria and viruses. And finally, as another example, we found out that in our B-cell cluster, we were able to follow the overexpression of CD74 and MZE1 that are respectively responsible to regulate B-cell transcription and other B-cell function. So this gave us a lot of confidence in our approach of clustering the cells and be able to label them correctly in our single-cell proteomic data. As a conclusion for this talk, we were able to collect and prepare and acquire about 17,500 PBMCs in just a little bit over 2 months, combining NPOP and PISCO, and to get about 1,600 proteins per single cells. We successfully identified a couple of immune cell populations, and we're still working on it to polish this work. And I hope that you will stay tuned for what is coming next, because right now we're working on trying to see how aging through the immune cells, but also how the different patient responds to vaccination over time. And there is a way more patient in single-cell recovery. We're currently processing 6 more patients from this cohort. And also we really hope to deepen the protein coverage of our single-cell using the PSM tags that everyone presented to you today. So to conclude, I just would like to thank our funders, our collaborators, the CDC and Proteome Science. Obviously, everyone has PTIs that all these projects to work. There are also more projects in the immunity, so I hope that you guys will stay tuned for like the next results that we will share with everyone. And I thank you for your attention, and we take any questions.
All right. Thank you, Thibault. Any questions from the audience? I'll start with one. So this prioritized approach looked like it allowed we prioritize amino-related genes, proteins specifically, and in one effort was to characterize those cell types, right, with the clustering as you demonstrated. How applicable do you think this approach will be for other types of cells?
That's an interesting question. I think overall it's applicable to any cell type and any function. I think that the power of the P-scope approach is to decide what question you want to answer by prioritizing those set of proteins or those set of markers. So it can allow you to specifically identify some rare cell type by prioritizing the detection of protein associated with them. It can also allow you to answer other questions such as, we could have taken this protein by another tail and decided to prioritize any genes related to aging. Or, for example, if you are interested in a specific pathway such as inflammation, you could prioritize any genes related to inflammations to be sure to see them in every single of your cells and to be able to draw conclusion out of cells above cells.
I might preempt a common question is, what about PTMs? Can we prioritize those if we look for those in these data?
So yes, we can definitely prioritize PTM. I think that the challenge of prioritizing PTM at a single cell level is to be able to see them and to detect them. So, but in our data set, we have a couple of PTMs that we identify and that we're trying to make sense of as of now.
Great, thanks. Any other questions from the audience?
How do you think about dealing with small cells that might have really little amounts of signal? How do you think about dealing in head with that versus maybe sending? It might be hard to discriminate whether it's a failed cell or
Yes, that's a really good question. So one of this information that we can acquire from the cell one is definitely the size of the cell. So we can. And also we have a picture of every single of the cells, so that's something that we can analyze a posteriori, and also to correlate the size of the cells to the signals to be able to figure them in a better way. So definitely something is doable, but it's also just challenging the full acquisition, right? Because there is so much peptides and proteins that could try and detect per single cell, and the smaller they are, the more complicated it is to detect those peptides.
What do you think the best thing about doing immunology through mass spec is? What is in your opinion, what does mass spec offer that you think benefits studying immunology through that the most?
That's a really good question, and that's probably why I joined PEI. I think that in the immunology field, there is so many unanswered questions when it comes to signaling pathways, or when it comes to protein interactions. There is a lot of redundant receptors that either use different signaling pathways or the same signaling pathways. Protein modification, how some protein modification could lead to different immune responses, and so on and so forth. I think that there is so many unknown in immunology and so many answers that proteomics could offer.
Okay, if that's it for questions, let's thank Thibault.